Good afternoon. My name is Sandra De Maria. I'm Professor of Radiation Oncology and Pathology at Weill Cornell Medicine in New York City. This afternoon I have the pleasure of discussing with you the role of radiation dose and fractionation in the immunogenicity of radiotherapy. So I will first review briefly the data that radiation can enhance response to immune checkpoint blockers, and then focus more on our work on the abscopal effect and how it can be induced in tumors refractory to immune checkpoint inhibitors when radiation is added to the treatment, and spend most of the time discussing our new results on the molecular mechanisms that regulate the radiation dose dependency of the abscopal effect, which actually are just coming out today in Nature Communication. So we use a mouse model of aggressive polyimmunogenic breast carcinoma, breast cancer in the mice that is refractory to treatment with immune checkpoint inhibitors, to test the hypothesis that radiation could actually synergize and restore responses. This is the 41, a model of triple negative breast cancer. It grows when injected subcutaneously in the mice with a histology of a poorly differentiated breast cancer that is not infiltrated or has very minimal infiltration by lymphocytes. So it doesn't really engage the immune system, explaining why this tumor is not responsive to anti-CTLA-4 blockade. This tumor is also very metastatic, and within the first week, as the primary tumor starts growing, it sheds metastasis in the blood, and so the outgrowth of lung metastasis is really responsible for the death of the mice. So one can irradiate the primary tumor and measure development of systemically effective anti-tumor immunity by looking at lung metastasis control and survival of the mice. So over 12 years ago, in collaboration with Jim Allison, again we tested a blockade of CTLA-4 in this model, and by itself, blockade of CTLA-4 was unable to induce any CD8 T cell responses to a tumor antigen measured here by interferon gamma production. And radiation really did not stimulate any response as well. However, in combination, they very nicely complemented each other, and we could detect tumor-specific CD8 T cell responses, and only the combination of radiation to the primary tumor and anti-CTLA-4 was able to extend the survival of the mice, which was due to improved control of the irradiated tumor as well as the lung metastasis, and we went on to prove that this was actually mediated by CD8 T cells in depletion experiments. So for those of you who were present this morning at the educational session, there has been a lot of work to show that radiation can prime T cell responses against tumor antigens. Just very briefly here, I use this cartoon to summarize the concept that radiation induces an immunogenic cell death, leading to peak uptake of the tumor antigen by dendritic cells that become activated, migrate to draining lymph nodes, where they activate CD8 T cells against the tumor. The activated CD8 T cells can then go back to the tumor and recognize the surviving irradiated tumor cells and contribute actually to the tumor regression following radiotherapy. So there is a lot of evidence in preclinical models that T cells play an important role in tumor response to radiation. However, in many cases, abscopal responses have been more difficult to achieve, even in experimental models. And these are really the most important endpoint in a clinic. And here I just show you in a cartoon what we mean. When you irradiate a metastasis, you see not only the disappearance, the rejection of the irradiated metastasis, but also of other metastases, other lesions that are outside of the field of radiation. And this is really a very important effect, which has been rarely seen with radiation alone, but really very rarely, but has been seen more often in combination with immune checkpoint blockers in patients. And this is really one great example of this. This is a patient with non-small cell lung cancer that came to the attention of Silvia Formenti a few years ago. He had failed all line of chemotherapy. Basically, no other treatment options were left for this patient. And Silvia decided to test if she can induce an abscopal effect with radiation and anti-CTLA-4, as we had seen in the experimental models. She irradiated one of the many liver metastases with a non-ablative radiation dose of 6 grade times 5, and she gave the patient anti-CTLA-4 ipilimumab, 3 mg/kg, the dose approved for metastatic melanoma for 4 cycles. And a few months later, the patient showed complete regression, not only of the liver metastasis, but all the other that was irradiated, but all the other metastases in the liver, base of the lung, and bone. And I am happy to say that this patient remains tumor-free almost 4 years later, from actually more than 4 years later, from without further treatment. So how can we achieve this type of responses? And one important question we asked was whether the radiation regimen mattered. To do that, we use more stringent models of metastatic disease, where both tumors are actually present as palpable nodules at start of treatment. One tumor is irradiated, the other is outside of the radiation field, and is followed for development of abscopal effects. So we decided to compare a single dose of 20 gray to hypo-fractionated regimen of 8 gray times 3 and 6 gray times 5. So all of the 3 radiation regimen showed similar ability to control the irradiated tumor, but none showed induction of abscopal effects. Anti-CTLA-4 alone had no effect on either tumor, and also did not really improve tumor control achieved with 20 gray plus times 1 on both the irradiated side and the abscopal side. There was really no abscopal effect. In marked contrast, the 2 hypo-fractionated regimen induced, when combined with anti-CTLA-4, led to complete disappearance of most of the irradiated tumors, and also partial and complete abscopal responses on the non-irradiated side. So these data, which are now almost 8 years old, did not meet with a lot of enthusiasm in the radiation oncology community, because most people think, and I guess correctly so, that higher radiation doses are more pro-inflammatory and they can release also more antigens, so they should be more immunogenic. So we went on to study these the mechanism of this difference. And we used the same model with the two tumors, and we decided to test the highest single dose of 30 gray, which is really at the limits of what can be used in a clinic. Of course, it led to very nice control of the irradiated tumor, no abscopal effect by itself. And when combined with anti-CTLA-4, there was really no evidence of any synergy. The irradiated tumor did not show durable complete regression, and there was no abscopal effect. While with the hypo-fractionated regimen of a grade M3, we were able to see again, reproduce the prior results of a synergy with anti-CTLA-4 with complete durable disappearance of almost all of the irradiated tumor and very nice abscopal responses. And these effects were mediated by CD8 T cells, as we demonstrated in the depletion experiment. So a very talented postdoc, Claire Van Puyerbachs, decided to take on this project and investigate what was different in the gene expression in tumors that were treated with 20 gray or A grade times 3, and she looked 24 hours after radiation, and striking was a difference in this set of genes, which actually correspond to the type 1 interferon pathway genes that were selectively upregulated only in the tumors treated with A grade times 3 and not 20 gray. So she then tested whether this signature of type 1 interferon was exclusively coming from the stroma or could also come from the cancer cells by irradiating the cancer cell in vitro in absence of other components. And again, she's seen the same pattern, A grade times 3, upregulating a lot of the type 1 interferon genes. In fact, when she measured interferon beta release in a supernatant, the cells that were irradiated with A grade times 3 behave very much as virally infected cells that release interferon beta. Now, interferon beta, interferon type 1, is known to be required for recruitment and activation of a special subset of dendritic cells to the tumor. And these Batf3-dependent dendritic cells are key for development of anti-tumor CD8 responses in the mice that have been shown by prior work of Tom Gajewski and Bob Schreiber's lab. So to find out if there was a differential recruitment of this subset, the tumors were analyzed a few days after receiving radiation, and sure enough, A grade times 3 treated tumor showed a very nice and significantly higher increase in recruitment of these Batf3-dependent dendritic cells than the 20 gray radiated tumor. And most importantly, these dendritic cells were also very much activated as measured by expression of CD40, CD70. Now, to confirm that these dendritic cells were actually essential for the development of the abscopal effect in mice treated with A grade times 3 and anti-CTLA-4, the experiment was repeated in mice that are missing, they are deficient in this subset of dendritic cells, and this completely abrogated the response on both the irradiated side as well as the abscopal side, confirming that this subset is essential, and also that it requires expression on interferon receptor for this process to occur, because interferon receptor deficient animals were unable now to show the synergy between radiation and CTLA-4 again on the irradiated side as well as the abscopal side. So hypo-fractionated radiation can stimulate cancer cell intrinsic type 1 interferon, but how does this occur? And radiation has been shown to lead actually to accumulation of double-stranded DNA in a cytoplasm of the irradiated cells. So we look for this, and sure enough, cells treated with A grade times 1 and even more with A grade times 3, there was accumulation of double-stranded DNA, shown here by quantitation, and here by immunostaining with an antibody specific for double-stranded DNA. And surprisingly, this was undetectable basically in cells treated with 20 gray single dose or 30 gray single dose. Now, double-stranded DNA in a cytoplasm is a known strong activator of type 1 interferon, and it does so by binding to cGAS, which is a sensor which is very important for the cells to detect viral infections, actually. And this leads to activation of cGAS, which produces cGAMP and activates STING, which in turn activates type 1 interferon gene transcription. So the same cGAS and STING sensors were really responsible for interferon production triggered by the hypo-fractionated radiation, because selective knockdown or either cGAS or STING abrogated interferon beta-1 release in cells irradiated with A × 3 or viral infected, as well as interferon beta-1 gene expression and expression of another important gene in type 1 interferon pathway. Now, the key experiment was to test if this cancer cell intrinsic type 1 interferon production is essential for generating the abscopal effect in mice treated with radiation and anti-CTLA-4. So to do this, cancer cells that contain an inducible shRNA-mediated knockdown for either cGAS or STING or a non-silencing construct were injected at the site that is irradiated, while the contralateral tumor was made with cells that have a non-silencing construct. So only the irradiated tumor is disabled in terms of the ability to produce type 1 interferon through this pathway. So after the tumor was established, mice were treated with doxycycline to induce the expression of the knockdown, followed then by the usual treatment with A × 3 and anti-CTLA-4. These images are confirming the knockdown has been activated because there is an RFP downstream of the construct that can be detected. So cGAS knockdown in an irradiated tumor completely abrogated the ability of radiation in combination with anti-CTLA-4 to lead to complete regression of the irradiated tumor, and most importantly abrogated the abscopal effect. This was the tumors in this case were really unable to increase the recruitment of dendritic cells and even more markedly their activation. And we could not detect the development of tumor-specific CD8 response in either the tumor draining lymph nodes, so locally, or the spleen, so systemically. And the abscopal tumor that is infiltrated by CD8 T cells in mice that have a non-silencing construct, when the tumor is knocked down for cGAS, this infiltration is completely abrogated as well. Pretty much demonstrated that this pathway is essential for the ability of radiation in combination with CTLA-4 blockade to induce abscopal effects, and very similar results were obtained when the tumor was selectively knocked down for expression of STING. So this leads to a model where radiation, when used in a single dose of 20 gray, may induce some cell death, but if it is not capable of recruiting the right subset of dendritic cells, this will not prime CD8 T cell response, and so we don't see abscopal responses. In contrast, where radiation is used in a way that can lead to type 1 interferon production by the cancer cells, this leads to recruitment of Batf3-dependent dendritic cells, which then pick up the antigen, become activated, migrate to draining lymph nodes, prime the T cells, which then can go back to the irradiated tumor to complete tumor rejection, as well as mediate abscopal responses. Now, the next question to address is why cancer cells treated with high-dose radiation do not accumulate double-stranded DNA. So we went back again to the viral literature, and the Judy Lieberman group had shown that HIV during the transcription of the RNA into the reverse transcription of RNA into DNA generates a lot of intermediate pieces, basically of double-stranded DNA, that are ideal to stimulate SIGAS and STING and the type 1 interferon production. But this does not occur, and that's because there is this DNase known as Trex1, which digests this DNA that accumulates in a cytoplasm, and that's a mechanism of HIV that HIV uses to evade innate immune detection. Now, we did look to see whether Trex1 was expressed in our irradiated tumor cells. And here is again the accumulation of double-stranded DNA, baseline, high-dose radiation, A-gray or A-gray times 3, and here is Trex1 expression, and sure enough it was markedly upregulated only in the cells irradiated with high-dose radiation when there is no accumulation of double-stranded DNA. So to confirm that Trex1 is really capable of regulating type 1 interferon production, the cancer cells were transduced with a doxycycline-inducible Trex1, so we can force expression of Trex1 independently from the radiation dose that is used to irradiate the cells, and forced expression of Trex1 completely abrogated the accumulation of double-stranded DNA seen in cells treated with A-gray times 3, and abrogated the upregulation of interferon receptor and interferon gene. Conversely, when cells were transduced with a knockdown for Trex1, this restored actually the ability of 20 gray radiation to lead to accumulation of double-stranded DNA, shown here by immunostaining, and also to lead to upregulation of interferon. Now, forced expression of Trex1 completely abrogated the ability of A-gray times 3 to induce abscopal responses and complete regression of the irradiated tumor in mice treated with A-gray times 3 plus anti-CTLA-4, and this was true also in mice treated with radiation and anti-PD-1. And conversely, again, when cells had a Trex1 knockdown, it restored the ability of 20 gray radiation to induce abscopal effects in combination with anti-CTLA-4. This is tumors that are contain the non-silencing construct. Of course, 20 gray used once or twice is very good at controlling the irradiated tumor, no abscopal responses. When the cancer cells are knocked down for Trex1, it restores the ability of a 20 gray times 1 to induce abscopal effects, but this ability is actually better with 20 gray times 2, which again reflects the same that we have seen with lower dose radiation. The repeating the radiation seems to amplify this pathway, induce stronger abscopal effects. So is this A-gray the magical dose? So to address this, we tested several cancer cell lines, human and mouse, with different radiation doses to see which doses leads to expression of Trex1 that will decrease the accumulation of double-stranded DNA in a cytoplasm. And I show here the example with the mouse mammary carcinoma TSA. Again, very low radiation doses do not really lead to much accumulation of double-stranded DNA, but this seems to really plateau between 8 and 10 gray. Past 12 gray, it starts plunging, and this corresponds to the upregulation of Trex1 in this cell. So there is kind of a watershed dose when you reach 12 gray and you go above, where the induction of Trex1 brings down the immunogenicity of radiation. And we have found very similar data with other human and mouse cells with slight differences. Some cells actually we can see induction of Trex1 with 12 gray, in others or even slightly lower doses, and in others it occurs only when you go above 12-15 grays. But this is basically the model summarizing what I just showed you. Radiation that is used in a way to induce accumulation of double-stranded DNA can activate cancer cell intrinsic type 1 interferon production and lead to a very effective recruitment of dendritic cell and priming of anti-tumor responses. But once Trex1 is induced at higher doses, all of these radiation immunogenicity and ability to synergize with the immune checkpoint inhibitors is lost. Now, is this clinically relevant? And while this is something that we are currently actively investigating, the results of a trial that was done by Silvia Formenti, inspired by the patient that I have shown you early, with testing radiation and anti-CTLA-4 in non-small cell lung cancer patients that had failed prior treatment, showed that pretty nice results with close to a third of the patients that completed treatment showing either partial or complete responses, and all the responding patients were receiving radiation doses that are below the range that should trigger Trex1 activation, 6 gray times 5, 9.5 gray times 3, so very similar to the doses that we found effective in the mice. Here is an example of a patient which received the radiation. Again, he had multiple lesions in the mediastinum, lung, and supraclavicular node. He received radiation to this lesion here in the hilum of the lung, and with ipilimumab, and went on to become a complete responder at the day of evaluation after completion of the 4th cycle of ipilimumab. And interesting enough, we had a pre-treatment resection of one of his tumors that showed that he does express STING, and we know by actually RNA-seq that it also expresses cGAS. And this is important because there is in the literature evidence that some tumor can lose expression of cGAS STING. Now, we did not have enough tissue to test this in all the patients in this trial, but very interesting data was that when we looked at interferon beta levels in a serum of the patients shortly after completing radiation, there was really no change from baseline in the non-responder, while there was a very nice induction in all of the responding patients, suggesting that that's really a relevant pathway for this. In conclusion, the radiation dose and fractionation are critical determinants of the ability of radiation to synergize with immune checkpoint inhibitors. Radiation seems to mimic a viral infection of the cancer cells that stimulates cancer cell intrinsic interferon beta, and this is required to prime durable CD8 T cell responses to poor immunogenic tumors. And I think the most important kind of take-home sort of message from this is that tumor expression of cGAS and STING may really be critical for optimal immunogenicity of radiation, and so it would be is something we are looking at in trying to understand if it could be used to predict which patient may benefit from the addition of radiation to immune checkpoint blockers. And additionally, Trex1 induction can, we think, is a new biomarker that can guide the choice of the right radiation dose in combination with immunotherapy, which again may not always be the same for all tumor types, but having a marker to decide, I think, is going to be very useful to make progress in this field. And I just want to acknowledge the people that contributed to this study. Most of the data that I've shown you in a mice model is work done by Claire Van Puyer-Bax with some help from other members of the lab, in particular Julie Diamond and Yasmin Salfas. Silvia Formenti, of course, is my long-term collaborator in this line of research and the PI of all the clinical trial, including all the studies I've shown you. These are other collaborators that helped us in this project and our funding. And thank you for your attention.